A patient walks into a clinic with a stiff, toe-heavy gait, rigid posture, resting tremors, and a completely frozen, mask-like face. To any examining neurologist, this is a textbook presentation of Parkinson's disease. The standard response is to prescribe levodopa to restore the patient's motor control, but the medication has absolutely zero effect. The symptoms continue to advance, leaving the patient trapped in a rapidly stiffening body. The true culprit is the ultra-trace metal manganese. It breaches the blood-brain barrier, embedding into the basal ganglia, lighting up the globus pallidus on an MRI. A healthy body holds just 10 to 20 milligrams, roughly the mass of a grain of sand. That infinitesimal amount is an absolute requirement for your survival. Yet, if that footprint shifts by just a fraction of a milligram into the brain, it permanently destroys your motor control. To understand why human biology tolerates such a volatile element, we have to look back at primitive single-celled organisms. Manganese formed the core of an ancient biological engine called photosystem II, which absorbed sunlight and physically ripped water molecules apart. That specific chemical reaction released oxygen gas as a byproduct, directly generating the breathable atmosphere of early Earth. Today, human cells utilize that exact same ancient metal, but we park it deep inside our microscopic power plants, the mitochondria. Evolutionary design sequestered this highly reactive metal as a precise chemical tool to handle the volatile reactions keeping us alive. Inside the mitochondria, manganese stabilizes the enzyme SOD2. As mitochondria generate power, they spit out toxic superoxide radicals, molecular shrapnel that shreds cellular DNA. SOD2 catches this exhaust. Its manganese core instantly neutralizes the radicals, converting them into harmless water before the cell melts down. In the liver, manganese serves as the obligate structural activator for arginase, the terminal enzyme of the urea cycle responsible for clearing neurotoxic ammonia from the bloodstream. It also activates glycosyltransferases, the cellular machines responsible for physically weaving the structural matrix of your bones and cartilage. Without this trace metal, the mitochondrial genome degrades, your bone matrix deforms, and metabolism simply stops. Acquiring it is entirely non-negotiable. This flowchart maps manganese hitting the intestinal barrier. The gut absorbs only 5%, bouncing the rest into feces. The tiny absorbed fraction travels to the liver, which dumps excess into the bile. But manganese and iron compete for the exact same cellular door, the DMT1 transporter. If iron deficient, the gut panics, wedging open millions of DMT1 doors. Because the gates are wide open and the system cannot tell them apart, the gut accidentally vacuums up lethal amounts of manganese. This flood completely overwhelms the liver's capacity to excrete it. A common, mild nutritional deficit, like low iron, inadvertently weaponizes the gut, turning a standard diet into a systemic neurotoxic flood. Occupational hazards bypass this gut filter entirely. Industrial steel welders inhale manganese dioxide dust, which crosses from the lungs straight into the bloodstream. Inside massive foundry environments, the heavy atmospheric particulate loads easily circumvent a worker's natural digestive defenses. In a medical setting, intensive care patients receiving long-term total parenteral nutrition, or TPN, face a similar vulnerability. The nutritional fluid contains trace metals that are pumped directly into the vein, completely bypassing the gut and liver. Once free manganese escapes into systemic circulation, it targets the globus pallidus. This perfectly mimics Parkinson's, but the mechanics are inverted. In Parkinson's, the sending dopamine factories degrade. With manganism, the heavy metal floods the neural gap, generating massive oxidative stress. Dopamine combusts, and that oxidative fire physically burns away the postsynaptic receiving towers. This explains why levodopa completely fails. The drug forces the presynaptic factories to pump out replacement dopamine, but in manganism, the receiver towers are gone. The signal has nowhere to go. Because the physical structure of the receiving network has been eradicated, the severe loss of motor control and the resulting stilted gait become tragically permanent. Catching this toxicity early requires a simple blood test, but when clinical pathologists run these samples, the results frequently come back as false positives. The medical team accidentally poisons the sample themselves. Standard intravenous needles are manufactured from stainless steel, which structurally contains manganese. The physical friction of blood rushing through the hollow core leaches trace amounts of the metal directly off the steel and into the sample. To prevent this needle nightmare, phlebotomists must draw blood using specialized plastic cannulas. They pull 5 to 10 milliliters into a waste tube to flush away any metallic fragments before drawing the real sample into a specialized acid-washed royal blue top tube. Even with the right tube, physical handling poses a severe risk. Red blood cells hold up to 20 times more manganese than the surrounding plasma. If the syringe is pulled back too forcefully, the mechanical shear stress pops the cells, releasing their internal payload and entirely ruining the test. Standard medical tools provide essential care while simultaneously contaminating the ultra-trace measurements required to assess the human body's internal metal burden. Once a pristine sample reaches the lab, scientists test it using inductively coupled plasma mass spectrometry. The machine injects the blood into an argon plasma torch burning at 8,000 Kelvin, completely vaporizing the sample down to its bare atomic ions. The machine acts as an ultra-precise scale, isolating and counting atoms based strictly on their mass-to-charge ratio. For manganese, the target atomic mass is exactly 55. Inside that inferno, other basic elements from the blood violently clump together. Nitrogen collides with argon, sodium fuses with sulfur. These random molecular clumps weigh exactly 55 atomic mass units. These molecular clusters act as perfect isobaric imposters. The machine's detector indiscriminately logs them as manganese, fabricating massive toxic overloads on the final report. The laboratory solves this physics problem using kinetic energy discrimination. Scientists install a collision cell directly in front of the detector and flood it with pure helium gas. As the microscopic stream enters the cell, the bulky imposter clumps constantly crash into the ambient helium atoms, lose their kinetic energy, and physically drop out of the beam. The true manganese atoms, being incredibly dense and aerodynamic, bullet straight through the barrier to hit the final counter. Achieving diagnostic accuracy requires isolating the exact mass of a single atom while dodging the chaotic fusion of entirely different elements. It illustrates exactly how challenging trace element medicine is for the modern clinical lab. Because the margins of error are microscopic, laboratories must maintain strict quality control, running daily calibrations against trace reference matrices distributed by the National Institute of Standards and Technology. This level of analytical rigor demands precise diagnostic stewardship. Heavy metal testing should be strictly reserved for screening occupational exposures or monitoring long-term intravenous nutrition, never ordered casually for routine idiopathic Parkinson's disease. But when an exposure is caught, the shared intestinal doorway offers a direct clinical intervention. By aggressively treating a welder's simple iron deficiency, physicians can physically shut the millions of open DMT1 intestinal gates, halting the accumulation of neurotoxic manganese. The behavior of this heavy metal reveals the complexity of our biological systems, from the mitochondrial shielding of SOD2 and the competitive DMT1 absorption gateway, to the postsynaptic receptor destruction, the physical leaching needle nightmare, and kinetic filtering of mass spectrometry. Our internal architecture operates as a calculated chemical network, where the permeability of a microscopic doorway and the integrity of a single analytical test determine the survival of our most sensitive motor pathways.